Okay, Karan, thank you for sparing the time to come onto the Health Path podcast. It's nice to connect.
Yeah, it's my pleasure, Alex. Thank you for having me.
No, thank you. I think you'll be glad to hear that actually I think you are the most requested guest for our podcast. We've had quite a few people ask to have you on. So people will be somewhat familiar with you, your work and what you do. But for those that aren't, are you happy just to share a little bit of an intro to yourself?
Of course, yeah. Well, number one, it's incredibly flattering to think that people are requesting. So it's my pleasure to come on and share my nerdy predilections with everybody. So I've got a lot of science nerd built up in here. So for those that aren't familiar, I'm Kiran Krishnan. I'm a research microbiologist by training, worked in a number of fields, including academics for a while, but then got focused in on the nutritional supplement space about now 20 years ago. And my first introduction into this space was I developed a clinical research organization with the whole focus of bringing good, affordable trials to nutritional companies. Clinical trials are very expensive, as some people might be familiar with, and it becomes really prohibitive for most nutritional companies to do good trials because of the cost. So my whole goal was, I think nutritional products should have trials, and many of them can be extremely beneficial. And with the right trial, you can actually prove some of that benefit. And so we wanted to create really kind of clever, smart, efficient, cost-efficient trials. That's how I got in. And then through that work, I got pulled into science advisory roles within companies, formulation, development, and so on, until I founded Microbiome Labs in 2013 with the sole focus of understanding the microbiome and finding tools to utilize the power of the microbiome to impact human health. So I've probably designed, run, and conducted over 50 human clinical trials. I've published, I think, somewhere around 8 or 9 of my own trials. So lots of experience in the research side and lots of experience in the formulation side.
Brilliant. Yeah, it must be a sort of a fascinating journey. And obviously our topic today is metabolic endotoxemia. So we're going to be doing a bit of a deep dive into gut health and the microbiome. And it sounds like obviously a very technical, sciencey, scary term. So are you happy to start just by kind of breaking this down for us? What is metabolic endotoxemia?
Yeah, so let's break down the terms themselves, because once people understand the terms, I think they'll understand what exactly is happening. So we'll start with the second term, endotoxemia. Endotoxemia, the ending of that word emia is associated with blood, right? And so endotoxemia means an endotoxin is now present in your blood at measurable levels. And there's a difference between an endotoxin and an exotoxin. So an endotoxin is one that's generated within the system itself, within our body in this case. An exotoxin is one that you get exposed to from the outside. And the advantage of an exotoxin is you can kind of get away from it. It's like a mold toxin. If your home is full of mold and you're getting a lot of symptoms from mold toxicity, step one is getting yourself either away from that home or remediating that home. The problem with an endotoxin is it's generated within and it's very hard to get away from it, right? And in this case, the endotoxin we're talking about is something called LPS, which I'll elaborate on in a moment. But that's the important part of the second part of the definition, is that it's an endotoxin that's leaking in essentially into your bloodstream. It's now found at high levels in your bloodstream, and that is very clinically consequential. And we can talk about what all it does once it's in your bloodstream. Then the first part of the word, metabolic, is referring to the process of digestion and consumption of food and so on. Because what was discovered was that this flooding in of the toxin into your blood, the anemia part, occurs at the highest level during the process of digestion and eating food. And so the biggest flood of the toxin entering your system is associated with the metabolic process of eating food. So that's why it's called metabolic endotoxemia. You'll also see it being referred to as postprandial endotoxemia. Postprandial simply means after a meal. So prandial being the derivative describing the process of eating, postprandial being after a meal. So after you have a meal, typically within 3 to 5 hours, you can actually measure this toxin in your blood and you could see the increasing levels of that toxin within your blood. That confirms postprandial endotoxemia or metabolic endotoxemia, or as many people know it, leaky gut. So leaky gut is the the more common colloquial term for that entire process.
Okay. So with sort of the postprandial side, am I right in saying that to some degree that's a natural response? And we're talking about kind of excess translocation of these endotoxins into the bloods?
So natural in the sense that it does happen at a low level in almost
Right.
everybody, right? But to define it as metabolic or postprandial endotoxemia, what you have to find is that there is a five-fold difference. Typically, is a minimum threshold that they create, a five-fold difference in serum or circulating endotoxin levels before the meal to the 3 to 5 hours after the meal, right?
Gotcha.
Now, in a normal person that may not have leaky gut, you might get a 1x increase, or you might get 1.5, maybe even 2 times increase. Your body can tend to deal with it pretty well. There's a number of mechanisms in our system to deal with the presence of this endotoxin in our circulation. But once you start getting to that 5, 6x level after every meal, it becomes really hard for the body to recover from it. And it sets the stage and the foundation for something called chronic low-grade inflammation, which becomes a foundation of chronic illness.
Yeah, so I mean, my brain's going in different directions here, but I guess, again, taking that step back, maybe coming back to this idea of LPS or lipopolysaccharides, are you able just to, I guess, share with people kind of what that is, what it means, and how that starts relating to maybe some of the conditions that we've associated with sort of chronic leaky guts?
Yeah, absolutely. So, lipopolysaccharide, or as you said in a much more fancy way, lipopolysaccharide, which I like. So the lipopolysaccharide is basically a constituent of the cell membrane structure of gram-negative bacteria. So every bacteria in the world can be designated as either gram-positive or gram-negative. What does that mean? Well, there's something called a gram stain. And so you can actually put a stain on the bacteria when you're looking at it in a microscope. And if the bacteria has a cell wall structure, then it'll pick up the gram stain. And when you look at it in a microscope, it looks gram-positive. So it changes the color of the bacteria. If it doesn't have a cell wall and it only has a cell membrane, then it doesn't pick up the stain. And when you look at it in a microscope, it looks gram-negative. It doesn't pick up the color of the stain, right? So bacteria will have, the gram-positive bacteria will have a cell membrane and a cell wall around it. And gram-negative bacteria only have a cell membrane, right? So that's the distinction between both types of bacteria. Now, the gram-negative bacteria have within the cell membrane this protein. It's a fatty protein. Actually, it's more of a carbohydrate than a fatty acid, a lipopolysaccharide. So saccharide, polysaccharide part of it is the sugar part, and then the lipo means fatty acid. So it sits in the membrane of the bacteria. The bacteria use it for numerous things like cell signaling, for binding to the mucosa, and a number of other things. And it sits inside the cell membrane structure of the bacteria. And it's perfectly fine when it's there in the cell membrane of the bacteria. In fact, the immune system uses it quite a bit in the mucosal structure, which is in the lumen of the gut. So it's still considered outside of the body. In that mucosal structure to kind of start training itself to understand which microbes are in the gut, right? And it's learning about the microbes based on what their lipopolysaccharide looks like. Now, where it becomes a problem is when those cells lyse or die or break open, and then the lipopolysaccharide is released, so now it's free floating by itself. And it's hanging out in the lumen, which is the tube part of your intestine. And as long as it hangs out there, it's totally fine. It moves out through defecation eventually, and it doesn't really cause any issue. When the gut is leaky, meaning the mucosal structure, which sits on top of the intestinal epithelium, which is the intestinal cells, the lining, if that's broken down and then the intestinal cells are leaky, meaning there's gaps in between them, then that lipopolysaccharide is allowed to leak past the mucosa, past the intestinal epithelium, and enter circulation. Once it enters circulation, it causes a massive issue. So 2 other things that are really important to mention. a good proportion of the bacteria in your gut, and these are your commensal normal bacteria, we're not talking about really bad pathogens, are actually gram negative, right? So that's a normal constituent of your gut. So having LPS in the gut microbiome is perfectly normal. Where it becomes abnormal is the barrier of your intestinal lining is compromised, and that LPS is allowed to leak through constantly. Now, what happens when LPS leaks through? Your immune system is designed to detect LPS because it uses that as a marker for the presence of bacteria. Because lots of infective bacteria, really scary infective bacteria, are gram negative, right? So over years, over millions of years, your immune system has come to recognize that if you have a high level of LPS in your blood, there's a huge danger because it signals bacteremia or septicemia, and it signals a blood infection coming. And so the immune system takes it very seriously, and it elicits this massive systemic inflammatory response in order to try to recruit and activate the immune system to the presence of this bacterial toxin in your blood. Because in the view of your immune system, if there's a certain elevated level of this LPS in your blood, it means that there's a potential of a flooding of your blood system with pathogenic bacteria, right? So every time LPS is in your blood, the immune response is that of blood poisoning or septicemia. So it's like a mini septicemia that occurs in your system. And the principal cytokines or inflammatory markers that increase because of the presence of LPS are interleukin 6, interferon gamma, and TNF alpha, right? So these are three critical inflammatory cytokines that get released systemically or localized, or in a local region if LPS binds itself in some local tissue within your body. And those are the quintessential inflammatory markers that start a very large cascade of inflammatory response, right? So imagine if your gut is leaky, every time you eat food, your body is going through a mini blood poisoning the 3 to 5 hour period after that meal. And that can occur and progress and remain for up to 2 weeks from a single meal.
Huh.
Right? That's how profound that immune response is, right? And then when you look at chronic illness, now what was discovered about 15 years ago, that most chronic illnesses were associated with something called chronic low-grade inflammation. That is a chronic elevation of interleukin 6, interferon gamma, and TNF alpha. And then there are other subsequent inflammatory markers that get activated because of these, right? Like interleukin 1 beta, for example, right? So this chronic elevated levels of these inflammatory markers seem to be associated with the vast majority of diseases that we deal with. So everything under metabolic syndrome, so diabetes, obesity, heart disease, Alzheimer's, polycystic ovarian syndrome, there's something like 60 conditions under metabolic syndrome. And then even other things like cancers and autoimmune conditions and all are associated with this chronic low-grade inflammation. The big question was 15 years ago, what is the source of this chronic low-grade inflammation? In the last 7 years or so, that question has been answered. And in fact, it's not been answered by peripheral functional medicine research. It's been answered by research by the NIH, by the American Diabetic Association, by the American Heart Association. Big mainstream research institutes have published dozens and dozens of papers showing that this chronic presence of LPS is the biggest source of the chronic low-grade inflammation, which then becomes the foundation for chronic disease. So that's the whole connection between it, and all of it starts with a dysbiotic microbiome, because the key thing that protects the lining of your gut and the integrity of the gut is the microbiome. We don't have in our own chromosomes capability of repairing and regenerating our mucosal structure and our intestinal epithelium. We count on very specific things from microbes in that environment in order to have a proper barrier.
Okay. So just to try and clarify one thing in this, am I right in saying that to some degree, an accumulation of these gram-negative bacteria, for whatever reason, maybe from dietary habits over a period of time, can induce leaky gut as well. So it could be sort of that actually these gram-negative bacteria induce leaky guts, translocate through, and then create the systemic inflammation, as well as potentially other factors impacting the integrity of the gut lining.
Yeah, so especially in the small bowel, right? So a lot of small bowel related disorders, things like SIBO, for example, or digestive issues, your primary digestive issues, IBS, inflammatory bowel disease, which then progresses into the colon. A lot of those start with issues within the small bowel itself. And what is pretty clear now that the small bowel typically is predominated by gram-positive bacteria in a healthy small bowel. When you start seeing issues like SIBO or really prevalent leaky gut, what you start to see is something called a taxa shift, where you get an increasing number of gram-negative bacteria in the small intestine. Once you start shifting to having a predominance of gram-negative bacteria in the small intestine, then you're going to start seeing really pervasive leaky gut being progressed throughout the small intestine. And the thing about leaky gut that's important to note is that you could have it in a few inch section of the small bowel, right, or the large bowel. But if it's not dealt with, if it's not healed, if it's not repaired and regenerated, then that leaky gut section will actually create leaky gut in adjacent sections. It's a progressive condition because all of the inflammatory response due to the leakiness in that section of the gut will actually bleed into adjacent sections and cause more inflammatory damage in subsequent sections. So, you might be 20 years old and have a few inches of your gut leaky and not really know it and never feel it, right? You feel perfectly fine. You can go to the pub and throw down a bunch of pints and eat whatever you want, not really feel it, right? But as you get older and that leakiness progresses, and there's larger and larger sections of the bowel that become leaky, then you start to feel the symptomology of that. And it may start as something as simple as irritable bowel syndrome, right? You start getting cramping, bloating, indigestion when you eat certain types of foods. You might start getting things like reflux. You might start getting things like allergies or intolerances to certain types of foods. You might start getting things like anxiety, or sinus issues. All of these things are related to that foundational shift in the bacterial taxa starting in the small bowel, and then that translates into the large bowel as well. Now, there's a couple of things that's really interesting. When you look at what would be a major source of either driving that taxa shift in the small intestine, going from a healthy gram-positive bacteria-dominant small intestine to a gram-negative, there's a few things that have been pretty well described. Number one is a contaminated oral microbiome. You've got a lot of gram-negative bacteria in your mouth, and the mouth is a major source of these types of gram-negative bacteria, like E. coli, for example. And so these, or Enterococcus, these tend to be predominant in the mouth, especially if you have an unhealthy, unhygienic mouth. Now you're swallowing those bacteria constantly. You're swallowing billions and billions of cells of those bacteria through saliva, right? And most of those are supposed to get killed in your stomach. Your stomach acid is supposed to act as a gastric barrier. But then if your stomach acid is compromised, either A, because of H. pylori infection, or the use of things like antacids, or the use of things like protein pump inhibitors for reflux and so on, then more of those microbes make it through in a viable state into the small intestine. Now, the small intestine has another protective function against gram-negative bacteria, and that's bile. So bile circulation, right? Bile acts as a very potent antimicrobial, and it does affect gram-negative bacteria and keeps those numbers low. However, bile is produced by the liver. So the moment your liver starts getting compromised from excessive drinking, lack of sleep, stress, all of these things, and then taking of drugs, things like, ibuprofen all the time, Tylenol, antibiotics, all of these medications and all that we take. And then, of course, contaminants in food like Roundup, right? Like glyphosate, so herbicides, pesticides, all of those things have to go through the liver and compromise the liver's function. So then you start shrinking your bile acid pool, and your bile acid pool becomes compromised. So that natural defense against the establishment of gram-negative bacteria in the small intestine also becomes compromised. And so now you've got a double whammy. You know, you've got a few years of compromised stomach acid, compromised liver, bile acid production, and then you start getting this taxa shift of gram-negative bacteria that starts to increase the leakiness in the small bowel. It starts to stop the movement of the small bowel, because one of the things that really compromises bowel movement, that's the peristaltic activity, the contractile activity in the bowel, is leaking in of LPS, the lipopolysaccharide. It goes up the enteric nervous system, up the vagus nerve, and it lodges itself in something called the dorsal vagal complex. The dorsal vagal complex is a checkpoint where the brain is sending neurological signals to the bowel to move, and also to send neurological signals to the bowel to elicit something called the migrating motor complex, which is electrical sweeping of the bowel. All of that gets compromised. So now your bowel is not moving the way it should. So food can remain for long periods of time in the small bowel and putrefy. You've got a compromised defense mechanism of stomach acid and bile salts. So now you've got an overgrowth of gram-negative bacteria. As you start getting an overgrowth of gram-negative bacteria, they release more LPS. As they release more LPS, you get more and more sections of the bowel becoming leaky. As you get more and more sections of the bowel becoming leaky, you're continually increasing your chronic levels of inflammation. That is the recipe for Western chronic illness. And again, you know, it's what's so interesting about it is when you look at the meta-analysis research, that's the studies of all the studies that are out there. And I do a presentation on this in a lot of medical conferences. You can take all of these seemingly unrelated conditions like reflux disease, depression, autoimmune conditions, acne. These are all seemingly unrelated because they affect different parts of the body in different ways. When you look at the root cause, they all have the exact same root cause. They all start in the gut.
Amazing. So I'm really glad you kind of mentioned the stomach acidity element of this, because I wanted to ask you around that, because obviously, as you say, the stomach acidity is meant to be one of those endogenous defense mechanisms against things like SIBO. Do you think that a sort of state of chronic stress that could induce hypochlorhydria or low stomach acid is enough also to have an impact on this kind of oral microbiome getting into the small intestine?
Absolutely. So there's good data showing that stress reduces stomach acid production quite dramatically, and also lack of sleep will do it as well. And lack of sleep does it to two different ways. One is there are, of course, circadian rhythms within the body, right? And the sleep part of it is the rest and repair component of it. And so as your cells that produce the stomach acid and so on, they get damaged over time just from doing the work that cells do. And in order for them to get adequately repaired so that you can continuously produce adequate levels of stomach acid, you need proper rest. And so it seems pretty clear that inadequate rest, so lack of sleep, disrupts hormone signaling, disrupts the signaling for stomach acid release. It disrupts the recovery of the cells, the canicular cells, and all of the cells involved in the production and release of stomach acid. And then stress compromises stomach acid release as well. So there's a number of factors there. And then, of course, the OTC use of compounds like antacids and so on. We've all gone and had a meal that was too big or drank a little bit too much. And the thing you reach for is antacids, right? To prevent that kind of bloated reflux feeling. And people get quite used to using those. And in fact, there's obviously you guys talk about a condition called SIBO, right? Small intestinal bacterial overgrowth. The use of antacids and the use of PPIs, the proton pump inhibitors, are considered a form of SIBO. PPI-induced small intestinal bacterial overgrowth is its own condition. And so, yes, absolutely, compromising stomach acid by lifestyle choices has a huge impact.
Okay, amazing. So I guess we're at the point where it all sounds a little bit doom and gloom, but I guess there's good news out of this as well, though, which is obviously there are various things we can be considering from a lifestyle, dietary, supplement perspective to restore balance here. So unless there's anything else, Karen, that you kind of want to just expand on there, should we kind of dive into some of the things that we can consider to support gut health with what you've been discussing?
Yeah, absolutely. So a couple of things to mention. There is a uncanny pervasiveness of this LPS, right? So we're finding now that LPS has spermicidal impact. So it is now associated with low sperm count and inefficient sperm in men. So it impacts fertility. It does the same thing in women. It impacts ovaries and estrogen cycling. It gets into deeper recesses of the brain, like the amygdala and the hippocampus, affects memory and processing capability. It impacts your skin. It has a massive impact all over your body. It is hard to find a condition or part of the body that you cannot find research showing that LPS it plays a role in a negative association with that, with conditions within that part of the body. And our focus became pretty zoomed in on LPS. And our focus became, how can you reduce the amount of LPS that's leaking into the circulatory system on a daily basis? Now, the reason I even thought about LPS a decade ago, because when we first started doing LPS-based research in terms of metabolic endotoxemia, really in the functional medicine industry, nobody was really talking about metabolic endotoxemia or LPS levels. And the reason it even occurred to me is because when you go through microbiology training and you start doing research, whether it's in graduate or postgraduate, we actually do a lot of studies on human intestinal cell lines, right? So they're called KCO2 cell lines. And one of the things you study, which is a kind of a basic study in microbiology, is what are the impact that the microbes have on the intestinal cells, and how do nutrients flow back and forth. And one of the ways that we try to make the intestinal cells leaky is by dripping LPS on it, right? So LPS has been used in research for decades now to create leakiness in intestinal epithelial cells, right? And so I remembered that from my academic research days, and I go, well, okay, so LPS must have a huge, profound role then in creating leaky gut in humans, actually, not just in a laboratory. And so we went down this road of trying to figure out how can we reduce that. And the good news is, which we'll talk about now, is you can absolutely reduce, eliminate, repair all of that, because it all comes down to an ecological issue within the microbiota. And you can fix the ecological issue through a number of behaviors. And when you fix that ecological issue, the ecology will fix the dysfunctional structures, right? And that's absolutely true at any age. So it's never too late to make improvements. It's never too late to repair those structures.
Brilliant. And I guess, it's what you say there. Obviously, more and more people are becoming aware of the gut-brain axis, but there's the gut-liver axis, as you say, the gut-skin, the gut-muscle, the gut-lung, it kind of goes on. So LPS is kind of, as you say, one of the mechanisms within probably all of these gut-something axes.
Mm-hmm. Absolutely, yeah. You'll find LPS as a culprit in creating dysfunction within all of those axes. You see that in lung disease. For example, people who have chronic obstructive lung disease, or people who are highly susceptible to pneumonia, tend to have more gram-negative bacteria in the lung microbiota and more LPS. And same thing with gut-brain axis. LPS gets into all these deep recesses of the brain. In fact, LPS can interfere with dopamine and serotonin binding, so it interferes with those receptors and their ability to bind the hormones. In the skin, you can find in the sebaceous glands an accumulation of LPS that actually creates inflammation in the sebaceous gland, which can lead to acne and other inflammatory conditions on the skin. So absolutely, and in fact, rheumatoid arthritis and other joint-related issues, you see an accumulation of LPS in those joints, which triggers an inflammatory response in those joints. And so same thing with diabetes. There's something called central insulin resistance, which happens irrespective of body weight changes. So you could be perfectly lean, you could be in your 20s, and you could develop type 2 diabetes. And there's more and more of that happening right now. And that's because of something called central insulin resistance, where LPS gets accumulated in the hypothalamic region, the J&K region of the brain, which then interferes with your brain's ability to read your blood sugar levels, right? Because it's the brain that's controlling the release of insulin, glucagon, and so on to maintain blood sugar levels within a safe region. And if the brain can no longer read your blood sugar levels because the signals to the brain are being disrupted by LPS from inflammation from LPS, then you develop something called central insulin resistance. And that's a major driver of the massive proliferation of type 2 diabetes that's occurring in the world.
Wow.
It's really crazy when you think about it. So look at two of the most scary degenerative conditions, right? Parkinson's and Alzheimer's, the prevalence of which continue to increase over time. There's millions of people that are going to be affected by these two conditions. Both conditions at the onset are triggered by the presence of LPS. In the case of Alzheimer's, it's the accumulation of LPS in the brain. In the case of Parkinson's, it's the accumulation of LPS in the central nervous system. And both of those things trigger inflammatory responses, which starts neurodegeneration. And that neurodegeneration is the beginning part of those diseases. So it's, and here's a crazy connection with Parkinson's, right? The first symptom of Parkinson's, which most people who study Parkinson's knows quite well, is chronic constipation, right? That's the first thing that occurs in Parkinson's. Why is that? Because remember, I talked about how the bowels stop moving when you have LPS accumulation moving up the enteric nervous system to the vagal nerve, or the vagus nerve, and accumulating in that dorsal vagal complex. So that's the initial part of accumulation of LPS in the central nervous system. It stops the bowel from moving, and then if that continues to progress, you start getting neurodegeneration, and you start getting symptoms of Parkinson's. So it's really, really profound how pervasive LPS is and what a significant role it plays in virtually every chronic disease that affects the vast majority of people.
Yeah, that is crazy. Okay, so moving on to this idea of the ecosystem and wanting to modify this as a way to modify the structural side of things. Where would you like to start with this?
Yeah, so there are some characteristics of what we would consider a healthy microbiome, right? And so really at the end of the day, the features of a healthy microbiome speak to the protective capability of a healthy microbiome against metabolic endotoxemia or postprandial endotoxemia. So let's break down what a healthy microbiome looks like. First feature of a healthy microbiome is increased diversity within the microbiome. The more diverse your gut microbiome is, the healthier you are. And in fact, studies show the longer you will live. The diversity in your microbiome will actually dictate longevity, because diversity in microbiome is inversely correlated with illness, chronic illness, infection, and so on, right? So that's one key factor. We want to increase the diversity of the microbiome. And after I describe each of these features, we'll go through and say how you actually do these things, right? So we need to keep in mind increased diversity in the microbiome is critical. The second part of it is the presence of strains called Keystone Strains, right? So these are organisms within the microbiota that not only protect the host through a lot of metabolic functions, but also supports and protects the rest of the microbiome. And that's why they're called Keystone Strains, like a keystone in the arch of a architectural design. It maintains the structure, right? And so these keystone strains maintain the structure of the microbiome. So some of those are like Akkermansia muciniphila, Faecalibacter prosnitzi, Bifidobacterium longum, Rhumnococcus. So there's a number of these keystone strains which are not only critical to the host, but also critical to the microbiome. And lots of studies show are directly inversely correlated with disease. So Akkermansia, for example, if you have high levels of Akkermansia, you're protected against everything under metabolic syndrome, right? So the more the Akkermansia is, the more protected you are. Same with Faecalibacter prosnitzi. You have high levels of Faecalibacter prosnitzi, you're protected against all kinds of inflammatory bowel conditions, starting with IBS, Crohn's, colitis, colorectal cancer, and so on, right? So these are really important species within the gut. These are all anaerobic species. You're not going to find most of them in probiotics. You can't really take them in probiotics because the moment they come out of the body, they're exposed to oxygen. Within 30 seconds, they die. So they're designed to be in the gut, in an anaerobic environment, and they play a really important role there. Then the third feature is the production of certain postbiotics. Things like short chain fatty acids are really critical to the integrity of the lining of the gut, the inflammatory and immune response in the gut lining, and also all of the metabolic response in the body. Right now, these keystone strains play a big role in the production of short chain fatty acids, right? But that's another separate thing we should look at as a key feature of a healthy gut. And the last thing is some sort of competitive force within the microbiome to keep opportunistic organisms at low, healthier levels, right? So it is not at all unusual to have pathogens in your gut. We all have pathogens in our gut. It's not at all unusual to have opportunistic organisms in your gut. We all do, all over the body and in our gut as well. But it's that diverse, healthy microbiome that keeps them in check. The moment the diverse, healthy microbiome becomes compromised, then they are allowed to proliferate. And when they proliferate, they start initiating metabolic processes that are not good for the host and not good for the barrier structure. So if we just remember that diversity in the microbiome, increased growth of keystone species, increased production of things like short chain fatty acids, and then a control mechanism for opportunistic and pathogenic organisms. If you have those four features within your gut microbiome, you've got a healthy gut microbiome and you're protected against leaky gut and LPS endotoxemia.
Brilliant. And is that last factor, I'm sure you've read it, there's a paper, I think it was 2016, just entitled A Healthy Microbiome, discussing kind of a lot of what you've mentioned there. And I think one of the terms they used was just resistant. And there's an overlap between a diverse ecosystem is a resistant ecosystem or a resilient one. But this idea of, I guess, competitive competition and, as you say, managing some of these potential pathogens or opportunistic organisms, is that sort of the same concept?
And that's exactly right. We call it the resistome, and we measure it on one of our tests that we use quite a bit, because you need to understand the resistance forces within the microbiome, because that dictates the stability of the microbiome, right? All in all, our achievement of health comes from building resilience within the system, right? And that's human nature. We as a species evolved to the top of the food chain and top of the evolutionary ladder because of our resilience, our ability to adapt into different environments and different conditions. One example I give people of that is how humans are really kind of omnivores, right? And we can adapt to different food sources and diets as needed. For example, a lion, as powerful and majestic as a lion is an obligate carnivore, right? If there's a drought and the lion's food, the wildebeest or whatever other mammals the lion's chasing and killing and eating is compromised, then the lion will starve to death. The lion can't go, I'm just going to dig for berries and roots and tubers and eat that instead of a wildebeest, right? The lion's going to starve. The same thing with the lion's prey, the wildebeest. If there's a drought and there's no vegetation, the wildebeest can't start hunting rabbits and eating squirrels and worms and so on. It's going to starve to death, right? But the human in that condition, if it doesn't have a mammal to eat, if it doesn't have, adequate vegetation to eat, humans will dig for roots and tubers. It can eat insects. We can eat berries and fruits and all kinds of different things to survive and make it through. We've got humans like the Inuit Indians that survive on 90+% of fat in their diet. Right now, if any of us went to and started eating their diet, would probably kill us. But they've got this evolutionary adaptation to being able to live in that region, in that freezing cold region, and eat fat as the largest part of their diet. And so, we have this resilience, and that's what I always dictate to people as, like, if you want to truly be healthy, you have to have resilience. What makes me cringe a little bit is when people say, oh no, I'm totally healthy as long as I don't eat this, and this, right, or do this, and this, right? They list like 15 things they can't do, eat, or experience. So that's not health. That means your microbiome and your system is very tenuous, and it won't take much to throw it into disarray.
So how, I guess, moving into this idea of we've got these four variables, What are some of the things that are within our control that we can, I guess, utilize to modify these 4 variables?
Yeah, so let's start with the diversity piece. One of the key ways of increasing the diversity within your microbiome is to increase the diversity of your diet, right? So the more diverse your diet is, the more different types of foods you consume, the more diverse your microbiome is. Most of what you eat actually goes to feed your microbiome, right? We digest and assimilate a very small proportion of the food that we eat. Most of it, think of it as feeding your ecosystem on the inside. And that's a big diversity in plant-based foods, diversity in animal-based foods, proteins, carbohydrates, fats, resistant starches. So just trying nuts and seeds. So trying to get a nice little diversity within your system. And again, that's kind of how we evolved, right? Humans did not, through the course of evolution, have the luxury of saying, I'm going to be vegan, or I'm going to be carnivore, or I'm going to be paleo, right? We just had to eat what was available. And we had to eat what we could find. And that was a big diversity in food sources. Some anthropological studies assume that or estimate that our ancestors consumed upwards of 600 different types of foods annually. You take an average Westerner and may eat about 15, 16 different types of foods, right? So increasing the diversity in our diet is huge. One simple way I always tell people to do is that, we all kind of have our habits of where we shop for our food and so on. If you go once a week to an ethnic grocery store, for example, in my region, we have this store called the H Mart, which is an amazing Asian market that has, vegetables and meats and fruits that we would never see at our Whole Foods and the regular grocery stores that we go to. And you just select one thing from that grocery store, whether it's a root, a tuber, a vegetable, cabbage of some sort, and then incorporate that into your diet for that week. You don't have to make a whole meal out of it. You can just chop it up, steam it, saute it, whatever it may be, just add it into your diet. And if each week you add one new thing into your diet and you try to maintain the exposure to those things, you're going to increase the diversity in your diet quite dramatically. So that's one thing you could do. The second thing is counterintuitive, but not feeding the microbiome, right? So a period of fasting actually dramatically can increase diversity within the microbiome. Your microbes within your microbiome are layered into different types of fermenters. You've got primary, secondary, tertiary fermenters that do their job at different times during the day and do their jobs during different parts of the metabolic cycle. So when you first bring in food into your system, all of those large macromolecules are going to be attacked by the primary fermenters, right? And the primary fermenters are going to then digest those and produce secondary metabolites that then feed a whole slew of secondary digesters of fermenters. And then they're going to produce metabolites that feeds another layer of microbes, right? And so that whole process of this cascading of feeding microbes where they feed each other, it's called cross-feeding within the microbiome, that takes long periods of time. From one meal, it could be 10 hours before the third layer of microbes are actually getting what they need in order to create their metabolic activity. And so if we constantly keep adding food into the system, then it triggers and selects for the primary fermenters for the most part. And then these guys down here at the base hardly get to actuate their metabolic activity, which means they don't get as much growth. They don't get as much representation, as much time to multiply and have their representation within the population, right? So a period of fasting to allow all of this cascade of microbes to go through their metabolic process can be very important. I myself do a daily intermittent fast. I do about a 16-hour fast almost every day, where I stop eating at some point in the evening, 8, 9 o'clock. Then I don't eat my next meal till around noon or 1 o'clock the next day. So I put most of that during the nighttime sleeping time. And then I've just kind of skipped the breakfast and not make my first meal till lunchtime or late lunch. So feeding a diverse, rich diet and then not feeding the microbiome, both of those things are important as well. Another way of increasing diversity is by getting outside. It is pretty clear in the research that the more contact you have with the natural environment, and I don't mean outside as in just walking up and down the sidewalk in your street, right? The more you can go into natural environments, whether it's in our area, forest preserves where you can go by a lake or you walk through, path in the forest preserve. Those kind of interactions dramatically increase the diversity in your microbiome because you'll pick up organisms from the environment, and those organisms from the environment will actually stimulate the growth of some of the endogenous species within your gut microbiome as well. Another thing is to reduce exposure of things that are hurting your microbiome, right? So when it comes to food, trying to go organic as much as you can so you get less exposure to things like pesticides and herbicides, which are known to be antibiotics in your gut, which bring down diversity quite dramatically. We did a study with King's College a couple of years ago where we showed that as little as 3 weeks of exposure to environmentally safe levels of Roundup reduced diversity in the microbiome very dramatically. And so imagine if you're getting that exposure every single day through foods that you're eating, it acts as a long-term antibiotic within your microbiome and shrinks your diversity over time quite dramatically, right? So the more you can go towards cleaner food, if you can grow a little bit of your produce in your own garden, that helps a lot because that's a cleaner source of produce. The more you can eat less processed foods, because processed foods will have preservatives, antimicrobials, and all that in there to provide shelf life, right? So the more you can reduce those things, and again, I don't want people to feel overwhelmed like they have to make these big radical changes. It's small changes over time consistently will create profound effects, right? So on the diverse food side I mentioned, just add one thing a week into your diet. On the fasting side, even if you can do 2-3 days of an intermittent fasting a week to start, that's great. If you can get outside once a week, if you can use it like a prescription, meaning, you prescribe yourself 30 minutes once a week to be outside in a natural environment, that in itself can have a huge impact. Then you can go to twice a week, 3 times a week as you build it into your routine. And then as far as your diet changes go, okay, I love eating cereal. I'm going to move from my regular cereal to an organic version, right? A simple single change like that. Or I love eating this category of food. I'm going to look for an organic version of it, like bananas, for example, or some other kind of food, right? So small incremental changes like that. Two more incremental changes that you can make will have a huge impact would be looking at your personal care products. Your personal care products like your lotions and soaps and shampoos and all of those things contain crazy chemicals in it, most of which have not been tested against the microbiome, but many of which will have a negative impact on your microbiome because of the nature of those chemistries, right? So if you can clean up one of your personal care products even, it's going to have a huge impact. For example, don't buy soaps that have antimicrobials in it specifically. You don't need that, right? Soap in itself is inherently an antimicrobial. You don't need soaps with added antimicrobials in it. Buy a clean shampoo, one that has mostly, food-based ingredients, organic ingredients. What I've been focusing on over the last 5 years is finding the cleanest lotion and the cleanest natural deodorant to use, right? Because those are things that will impact my ecosystem. So that's another thing. And then the last thing was your home environment. So your home environment does not need to be sterile. Most surfaces in your home do not need to be Cloroxed and sterilized and have that bleach smell, right? In my house, most of the surfaces we clean using a squirt bottle of water, and I put a couple drops of essential oil just for some smell, and that becomes the surface cleaner. Now, if you bring home a raw chicken, you get raw chicken juice on your countertop, you do want to sterilize that because that can be Salmonella, Campylobacter, and so on. So be smart about it. But most surfaces you do not have to sterilize. That actually has a long-term effect on your microbiome. There's something called the Finnish allergy study, which confirmed that in a long-term study, which showed that homes that use more sterilization, in terms of their cleaning practices, had kids that have higher incidence rate of allergies, viral infections, microbiome dysfunctions, and so on. Right? So trying to keep your home environment less sterile is actually good. So those are a few things you can do to improve the diversity and improve the ecosystem within your microbiome. To increase the growth of the keystone species, right? So let's just focus on 2 keystone species or 3 keystone species that are really important. Akkermansia. Akkermansia plays a really important role in the mucosal structure. Fecalum bacteria, which is highly protective of the gut lining. It reduces inflammatory response in the gut lining and stimulates the rebuilding of the gut lining quite dramatically. And then certain types of bifidobacteria like bifidolongum is really important in improving metabolic and immune response in the gut lining and also rebuilding that mucosal structure, right? So we just take those three to focus on. Akkermansia loves polyphenols. So if your diet is rich in polyphenols, or if you can increase polyphenol intake in your diet, then you're doing a great job. The good news is there's some really fun polyphenols like dark chocolate, red wine, a clean red wine. You know, if you can get Italian red wine is pretty clean. If you get California red wine, there's a good chance that there's some pesticides in it. But I think the Tuscan wines tend to be very strict on what they can use in the vineyards, right? And so, so those are fun polyphenols. But of course, lots of sources of polyphenols come from colored fruits and vegetables and so on. So Akkermansia loves polyphenols, and Akkermansia loves fasting. So you can increase Akkermansia in those two ways. Fecalum bacteria prosnitzi likes certain types of resistant starches or oligosaccharides. So if you're eating things like kiwi, the pulp in kiwi will feed Fecalum bacteria prosnitzi. So it likes these resistant starches and these fibers. Bifidobacterium is very similar as well. It really likes resistant starches and complex fibers like oligosaccharides. Now, to make all of that easy, we created this product called Mega Prebiotic. Mega Pre is formulated with these unique oligosaccharide prebiotics that have been clinically shown in published studies to specifically feed those those important keystone strains: Akkermansia, Fecalum bacteria, bifidolongum, right? So it's 1 scoop a day, and what we see is about 100-fold increase in those keystone species in a 3-week period.
Wow.
We published a study on that in August of 2019. So a very easy and simple way, and you add that to the dietary changes, you'll really be supporting your keystone species. Another way of increasing keystone species, and in fact, starting to seal up the gut, is with the spore-based probiotic. That's why that was our first focus with Megaspore, is we said we want to see if this product can actually create that competitive resistance within the microbiome. It can bring down the growth of some of those opportunistic organisms and then support the growth of other species within the microbiome, and then also seal up those tight junctions in the intestinal epithelium. And sure enough, we saw in a 30-day study in individuals that had severe leaky gut or metabolic endotoxemia, just by taking the Megaspore alone, not doing any of the other things we talked about, right? Just by taking the Megaspore alone, saw a 60% reduction in the amount of LPS leaking into the circulation every day, right? 60%. That's a massive reduction. And then along with that reduction of LPS, we saw all of those key inflammatory cytokines that I mentioned, interleukin-6, interferon gamma, TNF-alpha, all those reduced quite dramatically as well. And we saw a number of markers that indicated the improvement of the gut-brain communication also, which means that LPS is now being reduced in the brain stem, in the enteric nervous system, and so on. So that was a published study in 2017, and we've since done a couple other studies, one large one that's going on right now to further establish those scientific facts. So the probiotic, the Megaspore in itself, will reduce that metabolic endotoxemia quite dramatically. If you add in the Mega Pre to increase the growth of the keystone species, and then you do some of the things we just talked about from lifestyle changes and so on, you'll have a profoundly different gut microbiome over a relatively short amount of time, a 2-3 month period.
Amazing. So you actually reminded me, I did have a little question that came into my mind earlier in regards to the idea of LPS translocating kind of into compartments of the brain. And is that something that almost like people talk about a body burden of toxins? Is it sort of that if we can improve the integrity of the gut lining, the microbiome, this kind of naturally just starts to come back down?
Absolutely. It'll naturally come back down. You'll get that low basal level of LPS leaking in, but your immune system can typically take care of that. You won't get that massive flood, right? That postprandial flood of LPS. Yeah.
Mm.
And it all is dependent on those changes to the microbiome, increasing the diversity, increasing the mucosal structure, the tight junctions in the epithelium, getting those keystone species up. All of those are features of a gut that isn't leaky and isn't allowing endotoxemia. It's your microbiome that's protecting you from that endotoxemia.
Mm. Brilliant. Is there anything else that you want to kind of touch on from a sort of an intervention perspective?
Yeah, stress management is critical, right? We talked about how stress can compromise your stomach acid, which then, of course, allows more of these oral gram-negative bacteria to make their way into the small intestine in a viable state and start the process of taxa shifting in the small intestine. Stress does other things as well. Stress creates an increase, of course, of corticosteroids and release of things like cortisol, epinephrine, norepinephrine, and so on. What is so interesting is that there are opportunistic microbes in your system that are designed by evolution to read and detect those stress hormone levels in your system. And when they detect an elevated levels of those stress hormones, that's when they turn on their virulence genes. That's part of the reason why they're called opportunistic, because they don't turn on their virulence genes all the time. They look for certain opportunities to do it. They've learned over the course of evolution, when the stress hormone levels are elevated in the host, it means that the host immune system is compromised. And so that's their best shot at proliferating, right? So as you are undergoing stress and you're increasing the stress hormones, then you've got these microbes sitting in your system, including viruses, that start to proliferate because of the elevation of stress hormones. So when they start to proliferate, they cause dysbiosis. Right now, that resistance factor in the gut is becoming compromised because you've got a certain set of bacteria that are highly motivated to proliferate. Then as you start getting more of those bacteria growing, they actually induce more stress because that creates a condition in which they thrive, right? So some of the organisms that proliferate in the presence of stress can actually produce neurotransmitters that they send up the enteric nervous system, up the vagus nerve to the brain, to create more stress and panic response in the individual, right? That's why we see stress and panic disorders as a progressive issue, right? It's not that like, oh, I'm just kind of stressed all the time. It typically gets worse over time, right?
Mm.
And so that whole activation of the HPA axis, the hypothalamic pituitary adrenal axis, is done in two formats. One is our own stress triggers an inflammatory response. One of the first things that occurs when we get elevated in our stress levels is your macrophages and dendritic cells start sending out inflammatory signals, right? Because the immune system goes, holy crap, our stress levels are high. That means opportunistic pathogens are going to start proliferating. So in order to try to control that, we need to release more inflammatory markers to recruit the immune system, right? So with stress, inflammation goes up. As inflammation goes up, the gut becomes leakier and more gram-negative bacteria and more dysbiotic bacteria are allowed to grow. So it's a self-perpetuating cycle. So mindfulness and doing mindfulness work to control your own stress can be profoundly powerful.
Amazing. Yeah. It's all, well, it's unsurprising now how many of these sort of systems and pathways are kind of bidirectional, as you're kind of describing there.
Yeah, absolutely. They all work on a lot of these feedback loops, right? Because again, the entire body is connected. One of the big faults of modern medicine is putting different parts of the body in silos, right? As if the brain is not connected to the heart, as if the heart's not connected to your joints, as if your joints is not connected to your liver, because we've got these specialists in different parts of the body, and all they're doing is focusing on that one organ, and that creates this misnomer that disease is organ-based rather than systemic, right?
Yeah.
And systems biology is the most critical aspect of it. And all of this has feedback mechanisms. As I mentioned, a leaky component of your gut is going to perpetuate leakiness because it keeps driving inflammatory response that compromises this integrity of the barrier in adjacent structures. And as that barrier becomes more compromised, it increases inflammation even more, which makes even more leakiness in other parts of the body, right? So these are cycles that we create for ourselves. So imagine your typical person that's listening to this interview. They are likely stressed, maybe because of their job or their personal life or a combination of the two. In order to overcome that stress, they're doing things like going out later, having drinks, maybe doing recreational drugs, staying up, watching TV, doing all of these things that then compromise their ability to sleep and repair, and actually increases the physiological stress within their system. All of that, all the while, is compromising the gut microbiome. Their diet is shrinking. They're getting less exercise. They're getting out less. They're working at home a lot more in front of the computer. All of these things perpetuate the dysfunctions that we've been talking about, right? So what we need to do is be very prescriptive and add into our routine small little things that are going to help us break that cycle. Because everything is this continuous cycle of dismantling of our system, right? And all the things we talked about are small. You could take a probiotic every day. That's easy. Take one, of course, that like the Megaspore that's been studied in leaky gut. You can take a prebiotic. You can increase your diet, the diversity in your diet quite easily. You can download an app and listen to a meditation 5 minutes before going to bed. And do that as the first thing you do when you wake up for the first 5 minutes of the day, right? Those are simple things you can do to just start making a change and break that cycle.
Yeah, no, I think that's so helpful, and hopefully helps people kind of just understand a little bit around what's going on in some of these situations as well. I guess one final question, Kiran, if that's okay, just because we're going to have a lot of people listening who are struggling with SIBO, and there is not misunderstanding, but there are certainly some people who are worried about taking a probiotic or a prebiotic when they have SIBO. So is there anything that you can just share quickly from that perspective?
Yeah, I've done a lot of lectures on SIBO, and I've done a lot of investigation on the pathophysiology of SIBO. I think one of the biggest issues with SIBO is the fact that SIBO in itself is labeled as a condition. And in actuality, we look at the pathophysiology of how it comes about. SIBO is not a condition of itself. It is a symptom of a larger problem, right? SIBO arises because of a lot of the things we talked about, and it's characterized by that taxa shift to gram-negative bacteria in the small intestine. But that only occurs when things like your oral hygiene is compromised, when your stomach acid is compromised, when your liver becomes compromised, when you've got too much leakiness in the gut so your bowel stops moving. Stasis in the bowel is a key feature of SIBO, right? So all of these things happen, and then you start getting the overgrowth in the small intestine. It's not that the overgrowth just shows up out of the blue, right? Because we've got all of these natural protective mechanisms against having overgrowth in the small intestine, and it's a compromising of those components that allows for the overgrowth. And the same thing that allows for that overgrowth is the exact same thing that creates leaky gut, metabolic dysfunction, anxiety, stress, reflux, everything else, right? So SIBO is just one feature of that dismantling of our system. So I understand how people become nervous about using probiotics with SIBO, and there are many probiotics that just don't make sense for people with SIBO. But what you do need if you have SIBO is you need a probiotic that helps with leaky gut, because that leakiness in your small intestine will continue to drive SIBO, because one of the first organs, and the organ that takes the largest abuse from a leaky gut, from endotoxemia, is the liver. Right? So if you Google endotoxemia and liver, you'll see all of this research that shows that endotoxemia leads to liver damage. And in fact, it's likely the biggest driver of non-alcoholic fatty liver disease and non-alcoholic steatohepatitis, right? Which is increasing and going way past hepatitis C now. It used to be people who were going into end-stage liver disease were because of hepatitis C or alcoholism. Now, non-alcoholic fatty liver disease is the most prevalent liver disease that leads to end-stage liver failure. Your leakiness in your gut is compromising your liver. A compromised liver cannot produce adequate bile acids. Inadequate bile acids maintains an overgrowth of bacteria in your small intestine. That's the loop, right? And if your stomach acid is compromised, you're going to keep swallowing gram-negative bacteria from your mouth, so you'll never stop the inoculation within the small intestine, right? And so you have to address all of those things. And the Megaspore, one of the reasons why we use it so widely with Sibo is because it plays that important role in stopping part of that loop, in alleviating that leakiness in the gut, and allowing the liver to start functioning the way it is. We published a study in 2020 showing that when you take Megaspore for 90 days in people with elevated triglycerides, we're able to bring down triglyceride levels by 38%, right? That's huge. What are triglycerides? Triglycerides are liver fat. When your liver is compromised and undergoing toxicity, it produces triglycerides as a way of protecting itself, right? And so elevated triglycerides are synonymous with chronic illness. And so bringing down triglycerides means we're making an improvement. We were able to show, because we know that Megaspore is reducing that leakiness in the gut. Our hypothesis was, if it is reducing LPS endotoxemia, we should be able to see that in an improvement in liver function. And sure enough, we saw that with triglycerides. So it's, yes, I wouldn't take any probiotic. If I had SIBO, I would take the right probiotic, one that's actually dealing with the root causes that's driving my SIBO.
Fantastic. Brilliant. Kiran, I'm mindful of your time. Thank you so much for sharing all of your experience and knowledge. I know it's going to be a super popular episode, so thank you very much.
It is my pleasure. Thank you so much for having me. In my view, the future of healthcare and medicine comes from people being empowered with the knowledge on how their bodies work and what's impacting them. Everyone has to be their own best advocate, right? You can't just lean on your doctors to perpetuate health for yourself. You have to be your biggest advocate. And it's programs like this that provide people that empowerment and that information. So thank you for doing what you do, and thank you for having me on. It's a pleasure to be able to do this.
No, our pleasure. Absolutely.
Take care.